Welcome to EPG Pathshala. I am Dr. Deepshi Thakral from All India Institute of Medical Sciences, New Delhi. I will be discussing the module on basics of flow cytometry. At the end of this module, we will learn the following. What is flow cytometry? What can a flow cytometer tell us about a cell? How does a flow cytometer work? Its different systems, the fluidics, optics, electronics, and we will conclude by an overview of data analysis. To start with a couple of definitions, the word cytometry is made up of cyto meaning cell and metry meaning measurement. In simple definition, flow cytometry can be defined as measurement of the properties of a cell in a flowing system. Flow cytometry technology can simultaneously measure and analyze multiple physical characteristics of a single particle, generally cell, as they flow in a fluid stream through a beam of light, usually a laser. What is to remember here is the scattered light by these particles is then measured and is proportional to the different properties. So, how this scattered light is directly proportional to the relative size. The light in the forward direction, the forward scatter, is directly proportional to the relative size and surface area. The light scattered in the side is directly proportional to the relative granularity or internal complexity. And if the particle or cells are tagged with fluorescent molecules like fluorochromes, it reflects the relative fluorescence intensity. Shown here is an example of how the data derived from flow cytometry looks, and what you see in the plot is based on the size and granularity, the leukocyte population can be divided into lymphocytes, monocytes and neutrophils. So, the smallest population that you see in terms of the forward scatter is the lymphocyte, and the largest in size is the monocyte. The most granular are the neutrophils. Furthermore, the gate around the red dots that you see in the middle plot can further be characterized by molecules which are fluorescently tagged, antibodies against the antigens in this population, which is shown plot on the right, FL1 and FL2, which I will be describing. Now, how does a flow cytometer work? Listed here are the three main systems of a flow cytometer: the fluidics, optics and electronics. The main function of the fluidics is to transport cells to the beam of light for interrogation. Therefore, cells in suspension flow in single file through an illuminated volume where they scatter light, and the emitted light is either fluorescent or forward side scatter is collected by filters, and this filtered light is then converted to digital values which are stored in a computer. So, this is a bench diagram showing the layout or basic structure of a flow cytometer. The fluidics, where the sample is injected, the cells in single suspension are injected and carried by sheet fluid to the light, the laser, which is the source being laser beam. The laser beam excites these particles, and the scattered light in the forward scatter is collected by the detector, optical detector, and the side scatter by the PMTs as shown. And different fluorescent lights are collected by the mirrors and detected by the detectors, the photomultiplier tubes as shown. Now, the sheet fluid which carries these particles after crossing the beam, it either goes to the waste in an analyzer, or some flow cytometers are equipped with sorters, the element of sorting cells. And here is also shown in the picture is a sorter where, based on the the charge on the particles, they are deflected and collected for further analysis. And on the left is, once the data files are generated from the flow cytometer, they are analyzed on a computer. So, now we will go through each system of a flow cytometer in more detail, starting with the fluidics. The important things to know about fluidics is that fluidics functions to transport cells in a single suspension to the beam of light for illumination and interrogation. Single cell suspension is the key here. The clusters, if present in this stream, can result in erroneous measurements. The detector will not distinguish between a cluster versus a single cell. It will treat each cell or a cluster as a single particle. So another important thing is that these single cells, as they pass through the beam of light, should be optimally illuminated, and so for that they should be in the center of the beam. And this is achieved by the design of the flow chamber in which the single cell suspension is injected. The samples is injected into a stream within the flow chamber. So as the flow of sheet fluid accelerates these cells and restricts them to the center of the sample core, this process, when multiple flows with substantially different flow rates come into contact, resulting in centering of the core stream and the sheet stream, this process is hydrodynamic focusing. The sample core flows separate but coaxially in a central core within the sheet fluid without mixing with the sheet fluid based on the principles of laminar or streamline flow. In the illustration of this flow cell, a beam of laser light of a single wavelength is directed onto a hydrodynamically focused stream of fluid. The sample flow rate is controlled by differential pressure between the sample and the sheet fluid. The sample pressure is always greater than the sheet fluid pressure at any given time. The sample pressure regulator controls the sample flow rate by changing the sample pressure relative to the sheet pressure. A lower flow rate for optimal illumination, the stream transporting the particles should be positioned in the center of the laser beam. And this is achieved by injecting the sample into a stream of sheet fluid within the flow chamber, also called a flow cell. The design of the flow chamber allows the sample core to be focused in the center of the sheet fluid, where the laser beam will then interact with the cells. The flow of sheet fluid accelerates the cells and restricts them to a center of the sample core. And this process, introduction of a large volume into a small volume in such a manner that it becomes focused along an axis, is called hydrodynamic focusing. And when the conditions are appropriate, the sample fluid flows in a central core that does not mix with the sheet fluid based on the principles of laminar flow. In this illustration of a flow cell, a beam of laser light of a single wavelength is directed onto a hydrodynamically focused stream of fluid. The sample flow rate is controlled by differential pressure between the sample and the sheet fluid. The sample pressure is always greater than the sheet fluid pressure at any given time. The sample pressure regulator controls the sample flow rate by changing the sample pressure relative to the sheet pressure and has a dynamic range. Now shown in this example is a lower flow rate which decreases the width of the sample core and restricts the position of the cells to a smaller area, and therefore majority of the cells pass through the center of the laser beam, thus providing optimal illumination of the cells, and emission from the cells is also uniform. Therefore, a lower flow rate is generally recommended for applications where greater resolution of data is critical, such as DNA analysis. By increasing the sample pressure, the flow rate increases the width of the sample core. This in turn allows more cells to enter the stream at a given time, and therefore they are more off-centric. With a wider sample core, some cells pass through the laser beam off-center and intercept the laser beam at a less optimal angle and therefore result in less optimal illumination. This allows more rapid acquisition of the sample, but at the same time it compromises the resolution of the data, but is allowed for certain qualitative measurements such as immunophenotyping. And as described, this is achieved by the differential pressure between the sample pressure and the sheet fluid pressure. So how this fluidics works is based on the differential pressure system. The sheet pressure will set the sheath volume flow rate, and then the difference in the pressure between the sample and the sheath controls the sample volume flow rate, and which is adjustable. All this is achieved by the design of the flow chamber, which defines the axis and dimensions of the sheath and sample flow. It defines the point of optimal hydrodynamic focusing and also serves as the interrogation point. Now, this is a diagrammatic representation of a flow chamber showing the injector tip where the cells in single suspension pass through are accelerated by the sheath fluid towards the illumination by laser beam, and the scattered light is then collected. Now, two basic flow chambers are available. One is the jet-in-air, which is best for sorting but has inferior optical properties, and the other is the fixed alignment, which is the flow-through cuvette with excellent optical properties and can also be used for sorting. The major difference between the two is that in jet-in-air cytometers, the sample stream passes through a small orifice in a nozzle tip before being intersected by the light beam in open air. So, shown here is an example of a jet-in-air where the light collecting lens is focused on the outside the flow cell in open air. Therefore, fluidic systems in this case should be free of air bubbles and debris and properly pressurized at all the time. Now, because of the alignment is not stable, so it requires alignment on regular basis. The fluidics, the laser beam is focused through an achromatic lens to intercept the fluid stream at an optimal angle and position after the stream exits the nozzle. Now, because of variations in focusing and in the position of the stream, the alignment of this cytometer is less stable than that of a benchtop analyzer and needs to be optimized on a regular basis. Misalignment of the laser beam with the stream can result in uneven illumination of the particles and increased variability in the signals emitted. And this results in decreased precision. Most benchtop cytometers have the fixed sample pressure setting, which are fixed alignment, and the sample stream is pressurized upward through an optically clear region of the flow cell or cuvette. Particles pass through the laser beam while they are still inside the flow cell. So, to summarize, fluidics functions by transporting the cells to the laser beam, and now the next system of the flow cytometer is the optics, which is made up of two components. The optics is made up of the excitation optics and the collection optics. Excitation optics includes the source of light, which is the laser beam, and the lens to shape and focus this laser beam. And collection optics is for collecting the signals emitted, so, which has the collection lens and detectors and the filters to route specified wavelengths of light. Now, coming to the lasers. Laser provides both monochromatic and polychromatic light of covering a variable range from UV visible light to infrared and of variable power. These can be available with inexpensive air-cooled units or expensive water-cooled units. And they provide coherent light, and the most commonly used laser is the argon laser, which is for 88 nanometers, also helium neon for the infrared. Then there are diode lasers and solid-state lasers. Now, it is desirable that every cell passes through the energy profile of the laser in the same place and be illuminated by the same number of photons. The energy profile across any diameter of the laser beam is Gaussian or bell-shaped as shown in this figure. For lower power lasers, it is desirable that this transit point is at the peak of the laser energy distribution. Now, when the core stream is much wider than the cell diameter, then cells which distribute at random in the core stream pass through differing laser beam energy levels, resulting in measurement errors. Therefore, this problem is sorted by the use of cross-sectional lens and by reshaping the laser beam to an elliptical cross-section. This flattens and broadens the laser energy profile of the laser beam so that transit across the distribution reduces the positional laser energy that cells off-center experience, and therefore the problem is minimized. Now, an adverse side effect of this is reduction of the peak photon energy experienced by the cells, and this loss may affect the resolution of the instrument. Therefore, the overall energy of the laser can be increased. The collection optics include the optical channel, which is the path that the light can follow from illuminated volume to the detector, and then there are optical filters which help in optimization of these signals and carry them to the light detector, which can be two types, photodiodes and photomultiplier tubes. The two common detectors include the photodiode, which are low power, cheap, provide wide spectral range, and these are for detection of strong signals when saturation is a potential problem, and generally for forward scatter detectors are photodiode. And then there are the photomultiplier tubes, which are more expensive with limited spectral range and high signal to noise ratio, used for detection of fluorescence. So, whereas optical channels include the forward scatter channel, when a laser light source is used, the amount of light scattered by the cells or particles in the forward direction as traversed by the light is detected in the forward scatter channel. The intensity of the forward scatter is proportional to the size, surface area, shape and optical homogeneity of the cells. Shown is a representation of the how the scattered light is captured by the forward angle light scatter captured by the forward scatter sensor, which is a photodiode. When the scattered light is perpendicular to the axis of the laser light, direction of the laser light, it is detected in the side or 90-degree scatter channel. The side scatter is proportional to the internal complexity of the cells and reflects the granularity. So, shown as an example of both the forward scatter sensor and a side scatter sensor. The difference between the two is summarized here. Forward scatter tends to be more sensitive to the surface properties of the particle and is generally used to distinguish live from dead cells, whereas side scatter tends to be more sensitive to the internal complexity and therefore is used for distinguishing the granularity, granular from non-granulated cells. And the third is the fluorescence channel. The fluorescence that is emitted by each fluorochrome is detected in a unique fluorescence channel, which is the specificity of detection is controlled by the wavelength selectivity of the optical filters and mirrors, as will be described. So, understanding what is fluorescence and what properties of a fluorochrome are selection criteria for designing flow cytometry experiments for selection of these fluorochromes will be reviewed. Now, a fluorescent molecule absorbs light energy over a range of wavelengths that is characteristic for that molecule. This absorption of light causes an electron in the fluorescent compound to be raised to a higher energy level. The excited electron quickly decays to its ground state, emitting the excess energy as photons of light. This transition of energy is called fluorescence. The emitted light will always be of lower energy and hence longer wavelength than the exciting light. The energy may also be lost by non-radiative processes. So, shown as the lifetime of a fluorescent process, the absorption associated with the electronic transitions is about 1 femtosecond, and most of the fluorescent fluorophores have a lifetime of around 5 nanoseconds. A long lifetime reflects the narrow absorption peak, and a short lifetime excited states have a broad absorption peak. And these are some of the units which are used for measuring the characteristics of fluorochromes: the quantum yield, which is defined by the photons emitted by the photons absorbed, and the fluorescence lifetime is the time delay between the absorbance and the emission. Also, the energy difference between the energy peak of absorbance and the highest energy of emission is referred to as Stokes shift, and these help in determining for the selection of molecules. Now, when a molecule in excited state comes to resting state, it may release energy by non-radiative processes called quenching and eventually dissipate as heat. The range of wavelength over which a fluorescent molecule can be excited is termed as its absorption spectra and the range of emitted wavelengths is its emission spectra. Also, photobleaching is another process in which results in an irreversible destruction of an excited fluorophore. So, these are some of the properties of fluorescent molecules which help in selection and performing multiparametric assay, flow cytometry based assays, which includes the optimal excitation wavelength. The molecules should have high quantum yield, which reflects the efficiency, should be photostable, and the excited state of the lifetime of the molecules. So, in simple language, a molecule which is small, bright and stable will have minimal perturbation in biological systems and is preferred. Another range of fluorescent molecules available called the tandem dyes, these dyes are designed based on the process of fluorescence resonance energy transfer. Resonance energy transfer occurs when the donor does not emit light as photon. Instead, the emitted light is accepted by an acceptor molecule, but the key is the distance between the donor and the acceptor. It should be less than 100 angstroms. The energy transfer is non-radiative and results in the excitation of the acceptor, and so this allows spectral shifting of the emission spectrum. Therefore, it is used for spectrally shifting the fluorescence emission. Shown is a diagrammatic representation of an isolated fluorochrome where it is emitting light, shown with the green boundary. And if the distance between the donor and acceptor is greater than 100 angstroms, then it does not result in excitation of the acceptor. So, the key is the distance between the donor and the acceptor, and this spectral shift is used for designing tandem dyes, but these dyes are very unstable and light sensitive. So, this chart summarizes a few fluorescent probes available, the common ones. So, on the left is the name of the dyes, their excitation wavelength and the emission maxima. Most of these are excited by the argon laser 488, FITC, PE and PERCP, and then the helium neon is used for excitation of APC, cascade blue in the UV range. So, this is just a very narrow list. The available dyes are huge. Fluorescence overlap or spillover is the wavelength, the emission spectrum of a molecule is a range, and it peaks at certain wavelength and also spills into the other channel, and this spillover can be removed by compensation. Shown here is the overlap of FITC in the blue bell shape, where it is spilling in the PE-PMT, and vice versa, a very minimal spillover of PE into FITC. So, now the fluorescence. So, after selection of the fluorochromes and learning different properties of a fluorochrome, an ideal fluorochrome, the light emitted from the fluorochrome is detected in a fluorescence channel, and the specificity of detection is controlled by the wavelength selectivity of optical filters and mirrors. So, we will review various choices of optical filters and mirrors are available. So, this is a list of the options available: the long pass filter, the short pass filter and the band pass filters. A long pass filter allows the transmitted light greater than the cutoff, which in this case is 520 nanometers. The light below 520 is blocked by the filter, whereas a standard short pass filter allows the transmission of light less than the cutoff, which in this case is 575 nanometers. A standard band pass filter has a range. For example, in this case 630 by 20 has a range of 620 to 640. So, this filter allows the light in this range to be transmitted and the rest is blocked. Now, some special kind of mirrors called the dichroic mirrors are mirrors which allow this blocked light to be reflected. Now, these filters are constructed they are placed at a 45-degree angle to the light source, such that the light which would have been transmitted by that filter is transmitted, and the light which is blocked is now reflected at a 90-degree angle. So, shown as an example of a dichroic filter, the light source transmits the light at the cutoff, but because of the angle of this filter, the blocked light is now reflected at 90 degree. So, this is a dichroic long pass filter of 500 nanometer, which allows light of greater than 500 nanometers to be transmitted, and the rest is reflected. The light below 500 nanometers is reflected light. Similarly, a dichroic short pass filter allows light less than 600 nanometers to be transmitted, and the blocked light below 600 is reflected at 90 degrees. So, in this layout of a laser based flow cytometer, this is a single laser 4 PMT flow cytometer, shows the flow cell where the single suspension is carried by the sheet fluid to the laser light for illumination, and the scattered light is then collected in by the photodiode, which is for the forward scatter, not shown here, and the PMTs for fluorescence and side scatter. So, the placement of different filters and mirrors allows the scattered light from the fluorochromes to be captured by different PMTs based on their wavelengths. This was optics, and how this captured light now is coupled to the electronics and converted to the digital signals. We will look into that. So, this process where the light signals are collected and converted to electronic signals is mediated by photodetectors, which includes the photodiodes and the photomultiplier tubes as described earlier, and then they are assigned a channel number on a data plot. The photodiode is less sensitive to light signals than the PMTs and is used to detect stronger signals, or generally the forward scatter signals, and PMTs are used to detect mostly SSE and fluorescent signals. The voltage pulse is created as the particle enters the laser beam and starts to scatter light or fluoresce. Once the photons of light strike one side of the PMT, they are converted into a proportional number of electrons that are multiplied, creating a greater electrical current. So, the two steps in processing signals from detectors is pre-amplification and amplification. The electrical current travels to the amplifier and is converted to a voltage pulse, which is then converted to a digital number. The highest point of the pulse occurs when the particle is in the center of the beam and the maximum amount of scatter or fluorescence is achieved. As the particle leaves the beam, the pulse comes back down to the baseline. The size of the voltage pulse depends on the number of photons detected, the PMT voltage or the pre-amplifier gain and the amplifier gain. Signals are amplified by applying a voltage to these PMTs and creating a greater electrical current, or by increasing the amplification gain. Now, amplifiers can be set in the linear or the logarithmic scales. Log amplification is generally used to separate negative from dim positive signals, and linear is often used to amplify the scatter and fluorescent parameters. So, what is important to know here is that the instrument is set to trigger when a signal rises above a set threshold. An electronic threshold is defined as threshold which is used to remove the noise from the signal, and it is used to limit the number of events acquired by flow cytometer. Threshold is set on one parameter, generally the forward scatter, and the value is defined. Only signals with an intensity greater than or equal to the threshold are processed and sent to the computer. So, it is a cutoff. It is used to exclude debris from analysis, and it definitely affects the analysis dramatically. So, shown as a representation of setting the threshold, how the signals below the threshold, which is considered as noise, is removed, and only the signals which are above the threshold are taken as positive signals. And how, by changing the threshold setting, the debris from the cell population can be removed is depicted here. So, the difference between linear and logarithmic amplifiers is shown, where a linear amplifier is generally used for measuring the forward scatter, because this signal is very strong. It has a narrow dynamic range, and a logarithmic scale has a wider dynamic range, where it expands the scale of the weak signals and contracts the strong signals. And this is used usually for the measurement of immunofluorescence. And this is how an example of lymphocytes which are labeled with CD8 FITC in a logarithmic versus a linear scale. So if you see in the diagram on the left, the two peaks that are shown depict the CD8 negative population and the CD8 positive population, and there is a clear distinction between the two peaks of negative and positive. The same data, when seen on a linear scale, will just expand the region in between the two peaks, and the two peaks are off scale. So this is how the amplification in the logarithmic scale allows clean data analysis. So once this data from each measurement for each event is referred to as a parameter and is acquired as a list of the values for each parameter and event as shown in a table. So here is the list of 3 events, for example, and 4 parameters that were acquired: forward scatter, side scatter, and fluorochrome FITC and fluorochrome PE. So for the first event, these are the values. We come to the last part of this module where we will quickly go through the approaches used for data analysis, how to make sense of fluorocytometric data. So these are some terminologies to remember: gating, and we will review each of them. The display of data can be single parameter histogram, dual parameter dot plot or contour plot, and also in 3D projections where the third dimension reflects the number of events. Now coming to the gating. Gating is a graphical boundary which is created around cluster of cells which are of the similar properties, making them homogeneous. So homogeneous population. And this boundary can be of different shapes, and this gating we will review now can be done in real time versus the stored data, and how we will establish these regions, various gating strategies. Could be quadrant analysis, forward gating, back gating, as well as for complex populations, mixture of populations, Boolean gating. So generally real-time gating is used for sorting cells, and software or analysis gating excludes certain stored data from a particular analysis procedure and is used for like analysis of signals after acquisition. Shown is a list of possible shapes of the gates that can be used for defining populations. How are these gates established? The approach can be objective or subjective and depends on the expertise of an individual. So in the top figure, where a histogram is defined by a linear gate, and in a scatter plot in the bottom figure, where like a rectangular gate is defining a population, blast population. So these are different shapes of gates that can be used for data analysis. It could be a freehand gate, a quadrant gate, and then also the frequency of these cells with other statistical, the mean fluorescence intensity, all can be also exported to a Excel sheet, and statistical analysis can be performed. So shown here is the light scatter gating, where again the distribution of leukocytes, as we've also seen earlier, how based on forward and side scatter, the lymphocytes can be separated from the neutrophils and monocytes in the 3 populations, and by gating out lymphocytes, they can be further analyzed based on fluorochrome markers. Now forward gating refers to a hierarchical starting from the raw data FCS file as shown in the left diagram based on forward side scatter. This can be further, a manual gate can be drawn to define the monocytic population in this case, and based on some fluorochromes where FL1, FL2 in this is termed as forward gating, where we are subsetting the population in a sequential manner. Back gating is where, based on the forward scatter, side scatter profile, we first look at the fluorescent expression of molecules on certain populations and then backgate it and actually see it on a raw data plot. For more complex populations, the Boolean gating is used, which is generally automatically designates subpopulations to be included or excluded from analysis. For example, a not Boolean gating creates a population that includes all the cells that are outside a selected gate. In this case, it's the not region 2, so it is taking the region outside R2 gate. And then the or Boolean gating, which creates a population that includes all the cells within 2 or more specified gates, and it is removing remaining events outside these 2 gates. And the AND Boolean gating creates a population of an area which overlaps between these 2 gates, as shown. The common region between R1 and R2 is an AND gate. Now, gates must have some area of overlap to create an AND Boolean gate. So, this was a general overview of data analysis, and the expertise of an individual will further, more practice is required to achieve gating. So, hence we conclude this module on basics of flow cytometry, where what we have learned are what a flow cytometer can tell us about a cell and its different systems, the fluidics, optics and electronics. Now, flow cytometry utilizes highly focused bright beams of light to reveal aspects of cells such as the size, granularity and also fluorescence as a fluorescence intensity of certain molecules on the cells, the cell components. Now, it also quantitatively analyzes cells and permits the measurement of these parameters at a really fast speed. The power of single cell analysis is further compounded by the ability to measure multiple parameters simultaneously on each individual cell and also isolate, purify and sort the desired populations. Thank you.